are featuring our speaker, Julia Alterman, who is at the RNA Therapeutics Institute. So we'll get started. She'll have her talk, and then we'll have a question and answer period following her talk. You can ask questions using the Q&A feature, and we will moderate those at the end of the session. So I'll just pass it over to you, Julia.
Fantastic. Thank you so much. I'm really excited to be here today, and thank you for inviting me to give an OTS webinar. Today, I'm going to be talking to you about some of our work applying hydrophobically conjugated siRNAs, both to dermatological conditions as well as muscular conditions. And so those of you that were at this past year's OTS in Barcelona might recognize some of the data from the first half of this talk, but then we'll have some brand new data that I'm very excited to share with you about some work we've been doing treating Fibrodysplasia Ossificans Progressiva. Okay, so we will start with the main technology that we're using to do these studies, and I think that this is probably a technology that the majority of the audience is familiar with, but just in case we have any new people to the field, I'll just briefly describe how siRNAs work. So siRNAs are these short double-stranded RNAs that once they cross the membrane and get into the cell can be loaded into this protein called Argonaute-2 or AGO2, and once loaded, the sense strand or the non-targeting strand will be discarded, and the antisense strand or the guide strand, which is complementary to a target mRNA, will be retained. And this is now called the RNA-induced silencing complex. And this complex can find its target mRNA, cleave its target mRNA, and the mRNA will be degraded, and it will no longer make protein. And so these can be readily used as therapeutics, except that you need to heavily chemically modify them, right? So RNAs by themselves can cause an immune response. They're difficult to deliver because they're very negatively charged. And you typically need packaging in order to deliver a naked RNA. But we have many different chemical modifications that allow us to turn these molecules into very potent therapeutics. And these include modifications to the 2' position with 2' fluoros and 2' O-methyls that help with stability and evade the immune system. We have internucleotide modifications like phosphorothioates, which also give us stability and can help you to kind of get into the cell a little bit. We have 5' vinyl phosphonate that protects the 5' phosphate, which is necessary for loading into RISC. And we can add many different kinds of conjugates in order to get unique delivery properties and bring you to a certain tissue or cell type of interest. And we call these informational drugs, and we love these informational drugs. They're very different from traditional small molecules, where sort of the PK properties and the PD properties, so its targeting and how it distributes throughout the body, are linked into one molecule. So if you try to change the structure of a small molecule drug, you're likely to change its targeting as well. But with informational drugs, we can actually design a chemical scaffold that gives us the stability and the PK properties that we want, the targeting properties that we want, and then we can plug in different sequences in order to target different genes of interest. So this is a very cool new platform and modality for the treatment of genetic disease. And I'm sure, as many of you know, there are a number of approved drugs using siRNAs as their modality. There's actually 6 now approved drugs, 5 of which require no formulation and depend on this sort of heavily modified chemistry and conjugate strategy. And so these are GalNAc conjugated siRNAs. You can see the structure on the right-hand side of the slide here. And these primarily target hepatocytes in the liver. And as you can see below, with a single injection of these GalNAc conjugated siRNAs, we can see 6 months to a year duration of effect. And so siRNAs are really promising new technology for achieving long-term silencing of problem genes. So in our lab, we're really interested in seeing if we can find ways to use this technology outside of the liver for extrahepatic delivery. We've explored a number of different organ systems. We've looked at the kidney, the heart, placenta, muscle, brain, lungs, eye, and skin. And so for this talk today, I'm going to focus on 2 of these organs. For the first part of my talk, I will focus on the skin, and for the second part of my talk, I will focus on the muscle. So the way that our lab does this is with a library of different kinds of conjugates that give you different delivery properties. So on the left, you can see that we have a number of different structures of hydrophobic conjugates. These include fatty acids, sterols, and vitamins. But we also have a technology that allows us to make these molecules larger and multi-targeting. And this is multivalency, which you can see on the right. And so we can have compounds that contain 2 siRNAs, 3 siRNAs, or 4 siRNAs. And so in addition to being able to target multiple genes in one unimolecular structure, they're also quite a bit larger, which changes the way that they move through the blood and distribute throughout the body. And then on the far right, on the bottom, you can see an amphiphilic conjugate that we call dendrimer, which can also give you some unique binding properties. And at this point, I just want to point out the chemists that have contributed to this library of conjugates. We have been building this library over the years, and so I just wanted to put their names up there so that you can see the chemists behind these new designs. So the conjugate that I'm going to spend the most time talking about today is docosahexaenoic acid. And so this is a 22-chain fatty acid. You can see the structure of it here on the left. And we've actually published a lot about this. I'm sure many of you have seen it. We have published a number of different papers looking at it in different contexts. And in fact, a very similar molecule to this one is actually in the clinic. I think it just finished phase 1 clinical trials by Comanche Biopharma, who is using it to treat preeclampsia in pregnant women. Okay, so now we'll move on to the first part of the talk, which is focusing on skin. And I'm going to talk about both intradermal delivery of siRNA as well as transdermal delivery of siRNAs, and I'll describe to you what I mean by that. So I want to thank everyone who participated in this project up front. All of the projects in our lab are very collaborative, and I think that's what makes science work so well. So I really want to shout out to all of these people who have helped along the way, especially to Aldina Therapeutics, who are supporting this project. You can see them here on the bottom right, but none of this work would have been possible without the pretty faces that you're seeing here. Okay, so there's actually a large burden of disease associated with the skin, right? Not only is it the largest organ in your body, but skin diseases are some of the most common of all human health afflictions and affect almost 900 million people in the world at any given time, according to the World Health Organization. And one sort of inflammatory target that's involved in a lot of these conditions is JAK1. And we decided to focus on 2 of these conditions, atopic dermatitis and alopecia areata, both of them affecting more than 30 million people in the US alone. So the first thing that you need to do when going after a new gene is synthesize oligos to screen, right? You need to figure out which sequence is going to be the best sequence to target your gene of interest. So we did just that. We screened siRNAs against both human and mouse JAK1, and we were able to identify a number of hits, especially one hit right here that I'm showing you on the left and on the right, which is 3033, that actually has cross homology between the 2, which is really nice. So if you want to use this compound to do your mouse studies and then eventually bring it forward into clinical trials, you're able to do that. And as you can see on the right over here, this JAK1 siRNA 3033 shows great dose-dependent silencing, both in human cells as well as mouse cells on the mRNA level. And if you look even further to the right at the bar graphs, you can see that we are also seeing silencing of JAK1 on the protein level. So it seems that this is a pretty potent compound. We also wanted to make sure that this compound is selective. We expect it to be selective because we designed it to only target JAK1, but we wanted to make sure that it wasn't impacting any of the other JAKs or TYK2. And as you can see here, this JAK1 targeting siRNA only silences JAK1 and does not impact the expression of JAK2, JAK3, or TYK2. Okay, so let's talk about how we might deliver these siRNAs to the skin. So as many of you know, it's actually very difficult to deliver large molecules to the skin because you have a stratum corneum barrier that protects you from exposure to large molecules. So we need to figure out a way to get past this stratum corneum barrier. One of the most obvious strategies we could use is just intradermal injection, right? So injecting the compound directly into the skin. But another strategy that we've been looking at is actually more of a topical, or what we call transepidermal delivery. And we do this with something called star particles. So these star particles are small microceramic stars with about 500 micron arms. And when rubbed on the skin, they will make small perforations in the skin. And this allows larger drugs like siRNA to seep into those perforations and get delivery to the different layers of the skin. So we really wanted to try and see if we could use both of these to look at siRNA functionality in the skin. So we actually used two different conjugates for this study. We used DCA, which I mentioned earlier, which is a hydrophobic conjugate. And we also used Dendrimer, which was an amphiphilic conjugate. And the rationale behind this was we thought that it was possible that the hydrophobic conjugate would likely give us more accumulation and potentially more silencing at the site of injection or at the site of application. But we wondered if maybe we could get more spread with something that was a little bit less hydrophobic, so that if you were doing injections, you could have that spread maybe to a greater distance outside the area of injection. Okay, so I will talk about intradermal delivery first. So what we did in this study was we injected siRNA conjugated to DCA or Dendrimer or an unconjugated siRNA directly into human ex vivo skin. And the first thing we looked at was which cell types does it accumulate in? And we were able to see, as you can see in this graph on the left, that it actually accumulated in most or all cell types in the skin. So you can see it in immune cells, fibroblasts, endothelial cells, keratinocytes, and melanocytes. We then wanted to see if it was actually silencing in the skin. So we looked at both the epidermis as well as the dermis. And you can see that actually all three silence: the unconjugated, the DCA conjugated, and the Dendrimer conjugated. But it appears that the DCA conjugated may give us a little bit more activity. And this seems to correlate with the slightly more accumulation that we see here on the left. We also wanted to look downstream of JAK. So when we're silencing JAK1, are we actually seeing the chemokines downstream silenced as well? Is this impacting the whole pathway? And so what we did was we actually stimulated with interferon gamma after treating with our JAK1 siRNA, and we looked for the expression of CXCL9, CXCL10, and CXCL11. And what we were able to see was significant silencing of all three of these chemokines as a result of silencing JAK1 siRNA. Okay, one of the questions when we're thinking about targeting the immune system is you really want to make sure that you are not delivering the siRNA that's going to be silencing a master immune regulator throughout the entire body. This can cause some unwanted off-target effects. And so if we're going to go intradermally, we'd like to make sure that the majority of the siRNA is actually going into the skin and that we're not getting significant leakage into other clearance organs. And so what we did was a number of 40 injections into live pigs for a total of a 5.2 milligram dose. And then we collected not only the skin, but other organs such as liver, kidney, spleen, and heart. And we looked at the accumulation of these oligos in all of these different organs. And I think you can appreciate from these graphs below with DCA on the left and Dendrimer on the right that we have significantly more accumulation in the skin after intradermal injection than we do in the other organs. So I think it's about at least 16-fold greater in the skin than it is in the liver. So this is promising. Next, I'm going to move on to transepidermal delivery with star particles. So this is a picture of the star particles on the left that was published by Tedros et al. In Nature Medicine. These were invented at Georgia Tech. You can see they're quite small when you look at them on the penny there, or on the human finger. And the idea is that you can rub these on the skin and they'll make these small perforations. So on the right, you can see these perforations stained with gentian violet. And that will be the areas where the siRNA can seep in. And so, one of the questions we always get is, how comfortable are these? Is it going to be uncomfortable to rub these sharp star particles on my skin? And actually, a number of healthy volunteers were asked to share how their experience with star particles were and whether or not it was tolerated. And what they found was that with high pressure, so rubbing with a high pressure with star particles was actually fairly well tolerated, with 92% of people saying that it felt comfortable and only 8% saying that it felt uncomfortable. And this is in contrast to their response to a hypodermic needle, where 42% said that it was uncomfortable and 58% said that it was comfortable. We also wanted to investigate sort of how use of these star particles changed in the mind of the user over time. So the first question was, how comfortable or uncomfortable would you feel applying these products by yourself or to yourself? And what they found at day 1 was that maybe a quarter felt very comfortable, half felt comfortable, and a quarter felt neither comfortable nor uncomfortable applying it to themselves. But as you can see, by day 10, the number of people that felt very comfortable increased. We also asked, how did the application of these skin products compare to your usual experience with other skin products? This includes body lotion, sunscreen, face wash. And as you can see in the beginning, people felt that it was different. Most people felt that it felt different than their other skin products, with a much smaller proportion saying that it felt similar. But then you can see as we get out to day 10, that only half of the people now say that it feels different, and half of the people say it feels very similar or similar. And so what this is all telling you is just that even if a new technology might feel a little odd or a little uncomfortable in the beginning, that people will get used to it over time and feel much more comfort applying it themselves. Okay, so now I'll bring you into our work with these star particles. So for our first experiments with these in ex vivo human skin, we actually decided to formulate the star particles in aloe vera, rub it onto the skin, and then apply the siRNA afterwards. And so that's what you're looking at here in these figures. You can see on the left, the star particles formulated in aloe vera. You can see in the middle, applying pressure to the skin to make small punctures. And then on the right, you can see we've used some tubing to apply the siRNA to the top of the human skin and avoid leakage of the siRNA to the underside of the skin so that we can be sure that we're actually delivering through the epidermal layer and not coming in from underneath. And so again, we wanted to look at which cell types these siRNAs accumulated in following star particle treatment. And same as before, it seems to accumulate in all different cell types: immune cells, fibroblasts, endothelial cells, keratinocytes, and melanocytes. And we looked at silencing. We were actually able to see significant silencing, both with DCA and dendrimer, in the epidermis and the dermis, while we do not see significant silencing at all when the siRNA is applied alone on top of the skin without any star particle treatment. So this suggests to us that it is, in fact, the perforations that the star particles are making that are allowing this siRNA to get into the skin and silence genes in the skin. Okay, so now I want to talk to you about an experiment we did actually with live pigs in vivo. And in this experiment, we actually formulated the siRNA with excipient. So we formulated about 3.5 milligrams of siRNA in excipient for a total of 225 milligrams with the star particles. So now you've got one formulation with siRNA and star particles all together in excipient. And you can rub this directly onto the live pig. And this is much more representative of what this kind of treatment would look like in the clinic. And what we saw was that we were able to achieve significant silencing both in the epidermis and the dermis 4 days after the siRNA was applied, suggesting that we may actually be able to use this kind of topical application of siRNAs to deliver drug to skin. So this is very exciting. Okay, so just to summarize the first part of this talk here, I've shown you that we have identified a great siRNA sequence targeting JAK1. I've shown you that that sequence silences significantly after intradermal injection in human skin. I have shown you that after intradermal injection, we get a great deal of accumulation in the skin of siRNA, but little accumulation in clearance organs. I've introduced you to the idea of star particles, and I've shown you that these star particles can allow you to get this kind of topical or transdermal delivery of siRNAs in human skin, showing silencing here. I've told you that these star particles are comfortable and well tolerated. And I have shown you that actually when we formulate them together, formulate siRNA together with star particles, that we can get activity in a live large animal model. And so what are the next steps for this project? Well, of course, we're working on a number of new targets, not just JAK1. So there are a whole slew of inflammatory targets involved in different kinds of dermatological conditions. We are working on some unique novel conjugation strategies to improve our delivery of these molecules with star particles. We actually have an upcoming study, so we'll have some data very soon on dose response, duration, and multi-dosing in pigs, applying the whole excipient siRNA and star particles together. So I'm very excited to see the results of that upcoming study. And we are working on new shapes for the star particles that might actually give us better perforations, more evenly spaced. And we're doing this with the Swiss Institute of Material Sciences, or EMPA. Okay, great. So now I'm going to move on to the second section, which is muscle. And today I'm going to talk to you about a disease we've been working on. We have been designing siRNAs to treat Fibrodysplasia Ossificans Progressiva. So the people that are involved in this project are shown in this slide here. I really have to give a special thanks. This has been a collaboration between our lab and the SHIM lab. They are experts in bone and muscular biology, and they have a lot of expertise in FOP, and we could not have done this without them. And I especially just want to point out Catherine Gross and David Cooper, who are leading the project on our side and actually just spoke about these results at an FOP conference and did a very good job. And so, yes, this is truly a team effort, and everyone deserves credit for these results. This work has been funded by the IFOPA as well as some funding from AAVAA Therapeutics. Okay, so for those of you not familiar with Fibrodysplasia Ossificans Progressiva, it is a devastating and debilitating disease. Essentially, patients, if they acquire any kind of injury to their muscle, they will grow bone where that injury occurred. So any kind of inflammation, any kind of injury, and they will grow bone in its place. And this causes people to acquire bone over their lifetime. This includes all of their skeletal muscle, diaphragm, jaw, so they are unable to feed themselves, and patients typically don't live into their 30s. There is currently no treatment for this disease, and we really wanted to see if there was something that we could do to help. So what is the cause of this disease? What's the pathophysiology? Well, actually, FOP is caused by a single mutation, a single nucleotide polymorphism in the ACVR1 gene. And normally, this ACVR1 gene will signal responding to BMP, which is a bone morphogenic protein, and it will form bone, okay? And then there is another version of this receptor, ACVR1B and C, which responds to activin A, which is an inflammatory signal. Now, in the case of FOP, there is a mutation in ACVR1, and where ACVR1 would normally not respond to activin A, in fact, activin A would be an inhibitory molecule if it bound to ACVR1. Now, with the mutation in the ACVR1 receptor, it responds to activin A as it would to BMP, and it forms bone. And this is what causes FOP. So we want to be able to, but activin A is going to have functions, necessary functions in the body that we want to preserve. So we want to be able to silence the mutant while leaving the wild type alone. So we've actually developed a strategy for this, and we published on this back in 2022, Conroy et al. In Nature Communications, if you're interested in checking out the strategy for how to target a single nucleotide polymorphism with an siRNA to achieve allele-specific silencing. So what you can see is here on the right, if you look over here, you can see that a single nucleotide is going to be different between the on-target and the off-target. That's the SNP. And so sorry, there's someone coming into the office. Okay, so here on the left, you can see that when we design siRNAs against the SNP, you will have the on-target will be, the on-target will have no mismatches and the off-target will only have one mismatch. Sorry about that. And what this means is that you will can get very good silencing of the off-target, but you may also get decent silencing of the off-target. And this is because one mismatch can be tolerated. So what we did is we actually introduced a secondary mismatch. So this secondary mismatch is going to be in both of the siRNAs. And so this means that now the on-target is going to have one mismatch where the secondary mismatch is, and the off-target is going to have two mismatches. And while one mismatch is typically tolerated, two mismatches is not. And so we expect or hope that we'll be able to maintain our on-target silencing and reduce our off-target silencing. And so here is the first screen where we have just the single mismatch, and we have moved the single nucleotide polymorphism to different positions within the siRNA. And you can see it at position 3, position 6, position 8, position 9, and position 15. And I think what you can appreciate here is that we were able to get beautiful silencing of the mutant ACVR1. And I think you can also appreciate, if you look over here at the y-axis, that we were also able to get beautiful silencing of the wild type ACVR1. So we're halfway there, right? We have an siRNA that's targeting the gene that we want to target, but it also seems to be targeting the wild type, which we do not want to target. So we moved forward with this siRNA targeting the SNP at position 6, and we added secondary mismatches. And now I think you can appreciate that we were able to significantly reduce, if not completely get rid of, the off-target silencing. So we chose this compound here called 6-9, 6 being the position of the first mismatch, the SNP, and the 9 being the position of the second mismatch. And so you can see here that now we have almost no silencing of the wild type ACVR1, and we have just very little very little expression left over of the mutant ACVR1. Okay, so then we wanted to see if this would work in patient-derived iPSCs, FOP patient-derived iPSCs. And so what we did was we treated these cells with either a control, so like a non-targeting control, or we used siRNA targeting ACVR1, the mutant ACVR1. And we looked at the changes in expression of the wild type gene and the mutant gene. And as you can see with the control, the majority of what's expressed is actually the mutant gene, and with very little wild type expressed. But then when we silence the mutant ACVR1, we can see a significant drop in mutant ACVR1 and much higher expression of the wild type gene. And so this is very promising. This shows us that in actual patient cells that contain this heterozygosity, that we can actually see specific silencing of one allele over the other. Okay, so now we wanted to look at some of the downstream functionality. So first we wanted to check in mouse, FOP mouse BMSCs. whether or not we could silence ACVR1, first of all, before we moved forward with any further mouse studies. And as you can see here on the left, with increasing amounts of this siRNA targeting ACVR1, you can see that we have significantly silenced ACVR1 mRNA in these cells. We then wanted to look at some downstream signals of ACVR1, so BGLAP, COL1A1, and IBSP, which are all osteogenic genes that are involved in osteogenesis. And you can see that with treatment with the siRNA targeting ACVR1, we get reduction in all of these downstream genes. Okay, we also wanted to know if this siRNA would work after activin A signaling. So when activin A, the ligand that will bind to this receptor and causes this aberrant signaling, can we add activin A in, activate this receptor, and still see an impact from silencing ACVR1? And so on the left, what I'm showing you is a downstream signal of the ACVR1 activin A binding ACVR1. This is IDI, which is an inhibitor of differentiation. And what you can see is that we get a significant upregulation of this IDI when we add ACVR1 to the media. But then when we use our siRNA targeting ACVR1, you can see significant silencing of that ID1. Additionally, we looked at calcium deposits. So you can actually stain for calcium deposits with alizarin red. And so we wanted to see if we could reduce the number of calcium deposits that were caused by activin A stimulation. And so we stimulated with activin A and then treated with siRNA targeting ACVR1. And we were able to see a reduction in these calcium deposits. And you can see this quantified here on the right. Okay, now we want to move into an actual mouse model of FOP. And so we used a heterozygous germline mouse model that is actually a great representation for what the disease actually looks like in patients. And this was provided to us by the IFOPA. And so as you can see on the left, here's our experimental setup. So what we do is we pinch injure the animals in the tibial muscle. We wait 3 days. And then we inject the siRNA, and then we waited a week and we looked at histology and distribution. And the siRNA here is Si3 labeled, so you can see it in pink and you can see the nuclei in blue. And I think you can appreciate that we get great delivery of these siRNAs to injured muscle, as well as heterotopic bone marrow and cartilage. We then wanted to see about efficacy of these compounds. And so again, we gave a pinch injury to the tibial muscle of these mice. 3 days later, we injected the siRNA targeting ACVR1. We waited 4 weeks this time, and then we did X-ray and histology. And what you can see here is we had a significant reduction in bone growth. So on the left, you're looking at an animal that just received NTC, and you can see that there is a large bone growth where the pinch injury happened. And on the right, you can see that there is a much smaller bone growth. And so this is quantified on the far right, but you can see that we've maybe gotten a 75% reduction in bone growth overall. But that's not good enough. We would like more bone growth, or sorry, less bone growth and more prevention of bone growth. And so what else can we do? What else do we know about the pathophysiology of this disease? Turns out that inflammation plays a very big role in this disease. So ACVR1 binding to activin A is actually an inflammatory signal downstream of a number of other inflammatory signals. And this is what happens when you get that pinch injury or some kind of injury to the muscle. So we really wanted to see what would happen if we combined this ACVR1 therapy with an siRNA silencing an inflammatory gene. And so there has actually been some publications that have suggested that the IL-1 family of compounds actually play a role in FOP. And so actually we wanted to look at whether or not IL-1 beta went up in FOP mice after pinch injury. So what we did was we pinched the tibial muscle and then looked 0 hours, 6 hours, 12 hours, and 24 hours post-injection. And then we measured IL-1 beta mRNA expression. And what you can see here, the black line is the wild type mouse and the gray line is the ACVR1 mouse. I think you can appreciate that there's a much higher upregulation of IL-1 beta in the FOP mouse than there is in the wild type mouse, suggesting that there is an increase in this IL-1 beta signaling in these animals. So the first thing we did, again, is to design siRNAs against IL-1 beta. And so you can see here, we have designed siRNAs against the mouse IL-1 beta, as well as the human IL-1 beta. And you can see here on the right that we were able to get beautifully dose-dependent compounds for both species. Okay, so now we wanted to see what would happen if we did a combinatorial treatment, right? So if we combined this ACVR1 mutant targeting siRNA with the IL-1 beta siRNA, could we maybe get some kind of synergistic effect? And so the same setup for the experiment is true. We did a tibial pinch injury, waited 3 days, injected the oligos, and then waited 4 weeks. And first, we looked at gene expression to ensure that we were actually seeing silencing with both the mixture as well as the individual oligos. And you can see here with ACVR1 expression that both the ACVR1 alone, as well as the combination of ACVR1 and IL-1 beta significantly reduced the signal of ACVR1. And over here on the right, you can see that IL-1 beta alone or along with ACVR1 significantly reduced IL-1 beta. So we've confirmed that these work and significantly silence the genes of interest. And when we look at the bone, I think you can appreciate here that while we do get reduction of bone with ACVR1 alone, and actually we do get reduction of bone with IL-1 beta alone, that we get a much more significant reduction of that bone when we combine the two with a dual injection. And so you can see that on the right here quantified, where only 8%, there's only 8% bone growth in the ones that receive the dual treatment, as opposed to 22 or 34% bone growth in the ones that received a single treatment, suggesting that this combinatorial approach might be a really good idea for FOP. Okay, so lastly, and we just got this data yesterday, but I could not wait to show it to you all, so I had to include it in the talk last minute. We have been playing around a little bit with hydrophobic multivalent siRNAs. I'm sure it's not a surprise to this group, considering the kinds of structures I showed you earlier, but we wanted to see if one unimolecular structure, including ACVR1 and IL-1 beta, would give us significant silencing of bone growth. So the same experiment, and this time we had 2 groups, one that was the mixture of the two, ACVR1 and IL-1 beta, and one that was an actual unimolecular structure containing both of those sequences. And what we were able to find was that both the mixture of the 2 siRNAs as well as the unimolecular structure were able to similarly silence or prevent bone growth. So this is very exciting as well. It's just yet another way to deliver these 2 molecules to patients. So now I would like to summarize part 2 of my talk. So we have shown you that we have identified a SNP-selective siRNA against the ACVR1 mutant receptor. We have shown you that we can use this siRNA to actually affect downstream osteogenic genes. We have shown you that these DCA siRNAs deliver very well to injured muscle, and that injecting these siRNAs into a mouse model of disease, we can see significant bone shrinkage with this ACVR1 targeting siRNA. We've also shown you that we identified a potential new target for FOP, and we designed some new compounds against this IL-1 beta, and we showed that the combination of ACVR1 with IL-1 beta actually seemed to give us a greater effect on the disease. So the next steps moving forward, we are looking at, again, additional inflammatory targets that are involved in this disease. We're doing some chemistry optimization for enhanced activity and duration. We're working with some hydrophobic multivalent siRNAs. And lastly, we're looking into doing this for systemic administration so that we can prevent bone growth throughout the body. So this is delivering siRNA systemically to muscle. And on this, I would just like to say that this is another project that's going on in the lab. So Annabelle Biskans, who was in the Corova lab a few years ago at this point, did publish data showing that docosahexaenoic acid can deliver systemically to muscle upon sub-Q injection. And then more recently, Ken Yamada has developed a novel chemical modification, extended nucleic acid, or XNA, that seems to give increased muscle efficacy and duration of effect. So we are combining these two strategies to try and get a more functional long-term solution for muscle silencing. And so if you're interested in hearing more about the work that we've been doing on long-term muscle silencing, I really encourage you to check out Hassan's either poster or talk at the upcoming OTS in Montreal, where he will be sharing all of that new data. And with that, I would be happy to take questions. And if I am not able to answer your questions today, feel free to reach out to me on LinkedIn.
Wow, thanks, Julia. That was an amazing presentation. So not only covering one topic, but you're even covering two topics within one presentation. So I really enjoyed it. So if you have questions for Julia, please use the Q&A function. But I, as always, will start with a small question. So you mentioned these mutations in JAK1. Causing several skin diseases. So can you elaborate a little bit more on the kind of mutations that you have? Because I kind of assume when you use an siRNA approach that these will be gain-of-function mutations. And always, I think there's always a second question. What if you lower this particular protein too much? What will happen?
Great questions. So it's not actually mutations in JAK1 that are causing the disease. It's actually more like autoinflammatory conditions where your body is just producing way too much of an immune response in a particular organ, right? And so these people are just having constant immune signaling in their skin. And so we need to dampen that immune signal. Your question of silencing it too much is an excellent one. I mean, it's one of the reasons why we really want to make sure that we're not silencing it systemically. So just because the skin is having an autoimmune or autoinflammatory response doesn't mean that we want to be treating, the liver or the heart the same way, right? It's just the skin that's having this issue. And so I think the key is to make sure that you really only are delivering it to the organ of interest and that you aren't delivering it everywhere else in the body. We can maybe look at different kinds of immune targets that we can go after that may have different kinds of side effects. But ultimately, in the beginning, it's just there is way too much inflammation. This is what's causing disease. And so we need to shut down some of these inflammatory signals.
Yeah, so to follow up, so you, I think during your presentation, you showed that you have at least that the siRNA is getting into different cell types within the skin. So do you also show lowering of JAK1? And what about the liver? What do you see in the liver?
So we have not done silencing in the liver. So the compound that we used to do the pig experiments was the 3033. 3033 does target human, it does target mouse, it doesn't target pig. So we were unable to look at silencing with that particular molecule. We have since developed an siRNA, and we actually published on it, that targets pig, human, mouse, and a number of other species. So we're using that for future pig studies so that we can make sure that we have both PK and PD and that we can look exactly at that. Is this minimal amount of accumulation in the liver actually leading to some level of silencing? So we will definitely take a look at that. And then what was the first part of your question?
I also forgot. So different cell types. So do you see also lowering in the different cell types?
Yeah, so we haven't separated out the different cell types and actually looked at silencing in those different cell types. But given the overall level of silencing that we see, I suspect that it is in a majority of them. But no, we have not actually isolated specific cell types and looked at the particular levels of silencing in those cell types. That's also on the list of things we'd like to do.
Yeah, and then so I think I really loved your delivery strategy with the star particles. I think it's a really amazing strategy, and I think it's also quite amazing that also the patients seem to not feel that it is very harmful for them. So I think that might be also quite nice. So how often do you need to apply this? Yeah, what is it? Get aloe vera star particle mixture to the skin. And how long will the effect last? So do you have anything on the duration of this treatment?
Yeah, that is a great question. So you saw that all the studies that we did here were for 4 days. So we did not go longer than that yet. But there's an ongoing study right now. I think we're going to be getting actually tissues from that study within the next week or two, where we're trying to ask that exact question. So we are looking out to at least a month. With star particle application, we're looking at loading doses versus no loading dose. So as you know, accumulation can contribute to the duration of effect. So maybe if you applied it a couple of times, then you could wait a month or 2 months, 3 months, who knows? So we're trying all of these things. We're trying different doses. We're trying different time points, different loading dose schemes to be able to answer that exact question, because we also want to know how often do you have to apply these in order to maintain silencing of your genes of interest.
Yeah, and then to think more about the clinical application. So I think most of the skin, I think you mentioned that the skin is the biggest organ of all, right? So how much do you need to cover your entire skin? Because I can imagine that it might be quite a lot.
Well, I think it would be hard to cover your entire body. I think for a lot of the conditions, or at least the ones we're initially going after, like atopic dermatitis and alopecia areata. So for alopecia areata, you would have patches where you'd lost hair on your head. And so you would be rubbing on those patches. And same with atopic dermatitis, you're not going to have it everywhere. It's not going to be on every square inch of your skin. There will be areas of flare-up that you can treat. I suppose if you really, if you really wanted to have a preventative treatment so you never got flare-ups anywhere, maybe you would have to do a more broader application. But I think we'll just have to see what that looks like when we get into the clinic.
Yeah. Just going to some questions in the chat. So I try to cover the skin part first, and then we move to the questions
Sure.
on the muscle later. So I have a question from Nick. Thank you for a great presentation. I agree on that. In the first part of your talk for the ex vivo human skin or the in vivo pig study, what was the gene silencing efficacy when using STAR plus non-conjugated siRNA?
I do not believe, I think it performs similarly to Dendrimer. I actually do think we repeated that experiment, but I didn't switch the graphs in. It performs similarly to Dendrimer. So when you're doing local delivery of siRNAs and you're just kind of allowing it to seep into the local area, unless it's got some extra hydrophobicity that's going to make it sticky, you know what? Actually, maybe that's not true. I think it is worse than Dendrimer. I'm sorry, I should have brought that data today. We have done it. I think it is worse than Dendrimer and definitely worse than DCA.
Yeah, because the Dendrimer did not give such a huge effect, right?
Yeah, it still worked, but not to the effect that a hydrophobic conjugate does, at least for this type of application. It has other uses.
Question from Jeroen. In the context of the skin, the star particles are relatively large. How does the effect of these particles compare to well-established microneedles? Additionally, have you looked at the immune response upon mechanical stress of the relatively large perforations of the skin by star particles?
Yeah, that's a great question. We haven't compared it directly to microneedling, but with star particles, it seems it's a little less invasive. You can just kind of rub it on your skin. We did have people use it for, I think, at least a week or a slightly longer period of time and then wait. And there is, in healthy individuals, right, who don't have an immune condition, there's no infection. It heals right away. There's not, you're not looking at like holes in your skin. These are micro punctures. And so, it heals very, very quickly, probably within a matter of hours or even less than that. And there was no sign of infection in any of the individuals who tried it. So we're not worried about it at this point. I think that, it is definitely a question with people you're going to be treating with an siRNA that's going to be blocking some immune signaling, right? And so we'll have to see kind of how that works once we get to the clinic.
So, another question. Did you do analysis to assess whether the interactions between DCA and the star particle has any problems with the release of the siRNA into the skin? So it might also counteract each other.
We have not done any specific studies, but I think that the ceramic is pretty inert. I don't suspect that you would get a whole lot of interaction with siRNA and the ceramic. But I suppose we'll have to look into that.
Let's see. Just quickly going to the chats, but it might be good to then move to the second part of the presentation. For the muscle part, did you do any biodistribution studies for the ones that you use for the muscle part?
I mean, we showed some distribution studies in one of the slides where we injected after pinch injury and saw that it went to injured muscle and heterotopic cartilage. In previous studies, we've been able to show that siRNA goes readily to all kinds of muscles. And I think both Annabelle and Ken will have published on this in their XNA and systemic muscle delivery papers. So yeah, we get biodistribution with systemic administration to every muscle group, including heart and tongue.
Yeah, so what would be your favorite strategy to move into the clinic? Would it be the subcutaneous injections or would it be IV injections?
That's a great question. I think it depends. I think there are other questions too about how we're going to deliver these molecules. So this is a IL-1 beta you're delivering systemically. So even though we like the idea of using them together and we like the idea of a unimolecular structure, you may want to be getting regular doses of siRNA, ACVR1, if you are a carrier and have FOP, but maybe you just want single IL-1 beta injections when you actually have an injury in acute circumstances, right? And so how we will deliver this, whether it's sub-Q or IV, I think we'll need to check and see. Currently, all of our data with systemic delivery of siRNAs to the muscle is sub-Q, which seems to work very, very well. So I would maybe argue for sub-Q. But there might be reasons why an IV would be a better delivery system.
Yeah, and you also, so I think you showed data from just regular siRNAs, but also you have the multivalent siRNAs. So are there any differences in delivery and also efficacy properties of these siRNAs? Because I can imagine when you have a bigger molecule that it might be more difficult to deliver or more in the right location. So
yeah, well, you'd be surprised. Sometimes actually the larger molecules deliver better. And one of the key things to kind of trying to make a multivalent oligo hydrophobic is remembering that you really are changing the level of hydrophobicity of that molecule, right? So instead of having one oligo and one hydrophobic unit, if you were to just add 2 oligos onto a hydrophobic unit, you've now significantly decreased its amount of hydrophobicity, right? And so how is that then going to impact the hydrophobicity you need to get into the muscle? Even if you would gain some PK properties from having a slightly larger molecule. So locally, they seem to behave exactly the same. We are going to get, at least on the silencing level. So we haven't yet, that data I just showed you is brand new. I got it yesterday. So we don't have any accumulation data yet. There may be differences in accumulation. I will keep everyone posted. But My suspicion is you won't see as much of a difference locally. A lot of things behave the same when you just inject them locally. Where you really start to see the differences between different chemistries is when you deliver them systemically, right? And then you get really different distribution profiles. And so we are also planning to inject those molecules either as a mixture or as a unimolecular entity systemically. And I think that will be where we really see kind of how the chemistry changes have contributed to our distribution properties.
It would be interesting.
Yeah.
So,
Yeah.
so how much? Because I know within your lab you have quite a few chemists that can do a lot of modifications on the siRNA. So,
Brilliant
did you do
chemists.
a lot on the modifications of the different siRNAs? And does it? How is the effect of the modifications?
Yeah. So for both of these projects, we have actually, we're kind of just using traditional modifications. So we are using, the old published DCA, traditional methylfluoro patterns, ones we've published before, phosphorothioate tail, 5' vinyl phosphonate. Nothing that I presented today, I don't believe actually include, except for the slide where I mentioned that at the end here, where I mentioned that XNA gives you increased activity in the muscle after a single injection. So I did not label my axes here, but this is an siRNA without XNA. This is an siRNA with XNA. So you can appreciate that XNA is going to improve that activity. So we actually have not included XNA on any of these compounds yet. So I suspect that we're still going to probably get a bump from that when we do that. So no, these that I presented today don't have any of our fancy chemistry, but we sure do have access to a lot. So I'm looking forward to incorporating those in second generation molecules.
Yeah, it would be very interesting to see.
Sure.
So I have a practical question.
Yeah.
So how do you quantify the amount of siRNA in the skin?
So we have a few different methods that we're currently using. So in the past, we have used PNA hybridization assay. So you lyse the skin. For those of you that have tried to lyse skin, it is not trivial. So we have actually a special probe or bead-based shaker, bullet blender that helps you to lyse the skin to really get that out. And then we have a PNA probe that is complementary to the antisense strand and then has a fluorophore on it. And so we can pull that antisense strand out and then we can run it on HPLC and we can quantify the amount of oligo that's there that way. We have also developed a stem loop PCR assay in the lab, which has been published before, and we've kind of adapted it and begun to use that a little bit in our lab. Recently, we've gotten some new equipment. So the plan with our new triple quad mass spec is to develop a much more robust way to actually measure the oligo that is in all of these tissues. So we're in between methods. I think most of the data I've shown here today was using the PNA method, though.
Okay. I have a question from Roberto about the SNP specific or the allele specific approach. So you mentioned that you can better have a second mismatch than that the actual mismatch works better, performs better. So why did you proceed with the mismatch 1 at position number 6? Is the first question. And have you ever tried a third mismatch so that you have siRNAs for 3 mismatches?
Yeah, so this is not our first time doing this. We have previously published on this strategy before. So the way that we picked the initial, I didn't show any dose responses today, but the way we have picked the initial mismatch is just looking at doing a dose response with all of the single mismatch SNPs. So I just showed you the screening data and then picking the one that's essentially the most potent against the gene, the target gene, the on-target gene, right? And you saw that there was we had some discrimination, but not a lot of discrimination. So we just wanted to make sure, let's be working with the most potent on-target, and then we can screw it up for the off-target, right? And so that's how we picked position 6. It actually turned out, I think it's the same position that the SNP ended up working in our previous publication too. So I think something about that may have made us feel like, oh, this is a special position. But ultimately, we decided it based on dose responses. And then third mismatch, you know, we could try it. We have had a lot of success with the secondary mismatch. So introducing a secondary mismatch really screws up the off-target targeting, right? I mean, you saw one mismatch is tolerated, two mismatch very much not tolerated. My suspicion would be if we introduced a third mismatch, and that mismatch would have to be against the off-target and the on-target, that we would maybe lose too much efficacy in the on-target to make it worth it. You know, maybe there's a certain place where you could introduce it where it wouldn't have as much of an effect, and we could explore that and see what that, you know, if there's anything interesting that we can learn there. But for now, we've been very happy with the kind of discrimination we can get with just the two, so we haven't really been exploring three, but now you've made me curious.
Might be nice to test in a small
Just to test, break it, just to see that I
just to see
can.
what's going on. And maybe another silly question. So people often compare the siRNAs versus the RNA skippers. So did you do some experiments with skippers as well?
No, we have done no experiments with gapmers.
So we still have lots of questions. So I'm pretty sure that I cannot cover all of them. So I will just ask two more questions,
Okay,
if that's okay to you.
That's fine.
Question from David. So do you think that a clinical trial is doable with the two siRNAs?
Yes. In fact, I know that it is because I believe that Comanche is using two siRNAs for their clinical trial against preeclampsia, targeting two different isoforms. And so it's a mixture. It's a little bit more of a pain when you're doing kind of early tox studies because you have to look at all different combinations of the two and figure out if that impacts anything differently. So there are a few extra things you have to do on the front end, but yes, it's definitely doable.
Yeah, so and then I have one last question. I think also it's also a question for moving into the clinic. So do you have any ideas how the combination of siRNA and star particle by the regulatory agency? Do you expect any safety concerns regarding the particles getting to people's eyes or children's, for example?
You know, we don't currently have any safety concerns. People are very familiar with kind of rubbing lotions or other kinds of body products or face wash on their face that has little, you know, pieces of beads in it for exfoliation, right? I don't think that it's too different from what people typically use. You know, obviously they would have to be careful. It's going to be a little bit sharper. So no, we don't have those concerns yet, but I think we'll have to think about, since we, you know, the hope or the expectation is that we're going to be able to let patients use these at home, right? Which would make it so much easier than coming in to get regular intradermal injections from your doctor. But we would need to make sure that that was safe and that people could be trusted to apply it at home and not hurt themselves. And so I think that that's, yeah, definitely something we'll need to think about.
Oh, with that, thanks a lot, Julia. I really enjoyed the presentation and also the Q&A. So if you have any additional questions to Julia, you can send the message to her LinkedIn, or you can just connect anyway. And I think that she will also be present at the OTS in Montreal.
Well,
Kind of assuming that. And then Erin had to go, but she left a small message that the next webinar will be next week Thursday, so same time, and that will be held by John Caput. So you can join us also next week, same time, same place. So thank you all for watching, and see you next week. Bye-bye.
great. Thank you, everyone.